My name is Morgan, and I'm here to tell you about the story of Ellie Electron. Now, Ellie Electron lives within an H2O molecule, or a water molecule, so she has 2 hydrogen friends and 1 oxygen friend. Our journey begins at the site of the chloroplast. Within the chloroplast, the stroma, or the yellow liquid that you see in the diagram, is found within the cell. Within the cell, you will see a stack of thylakoid membranes I have circled right here. This is a stack of thylakoid membranes, also called a granum. This is where a big light beam filled with photons is absorbed by the chlorophyll. When the light is absorbed by Ellie's hydrogen friends, they break up with her oxygen friends, and Ellie goes to join the electron acceptor. Now, the inside of the thylakoid is called the lumen, and in this space, we enter phosphorylation. In this diagram, you see the thylakoid membrane, which is made up of a phospholipid bilayer, the chloroplast, and the lumen. Ellie is now transferred to the P680 molecule. In order for Ellie to be transferred to the P680 molecule, 2 water molecules must be split. The oxygen we breathe is a product of this breakup. Ellie then travels down the ETC and into the B6F complex. It is in this complex where the photon gradient is formed. Ellie then travels to photosystem 1. In order for Ellie to make it to photosystem 1, the ETC provides energy for synthesis of ATP by chemioosmosis. In photosystem 1, energy from the ETC gets absorbed by ATP energy. ATP energy stands for adenosine triphosphate. And within this stage, another photon is added to photosystem 1, P700. P700 absorbs red light. And within this stage, electrons bounce from light harvesting complex, kind of like a trampoline, and Ellie loves trampolines. All light colors are absorbed, except for green, which is why plants are green. As we can see, Ellie travels up the green path. We cannot forget about the red path with our hydrogens. Our hydrogens travels to the ATP synthase, and after going through the ATP synthase, it turns ADP energy to ATP energy. Now back to Ellie. After photosystem 1, she gets spit out into FD. And FD converts NADP energy with the addition of one of our hydrogen friends into NADPH energy. The next stage in our journey is the Calvin cycle. Ellie now starts her journey through the Calvin cycle. The Calvin cycle captures carbon and turns it into sugar. We're going to think of it like a factory. We're going to start with CO2 molecules, or carbon dioxide molecules, and this is in phase 1, carbon fixation. Within this phase, we take air, or carbon dioxide, and mix it with ribulose biphosphate, or RUBP. This creates a 5-carbon chain. It can only make the 5-carbon chain with the initiator, called riboso. Riboso binds the C from CO2 to the RUBP to build a 6-carbon molecule chain. Now, the 6-carbon molecule chain will split into 2 PGA chains. PGA chains are made up of 3 carbon molecules. And during this whole process, the carbon dioxide loses its O2. The next stage is the reduction phase. And in this stage, we convert 3 PJA to PGA with the help of ATP plus and NADPH, also known as energy from our light reactions. Now we're going to think of ATP plus as the delivery man, as it delivers energy to the molecules. And we're going to think of NADPH as it fixes the H molecules to each 3-carbon chain, kind of like a doctor. By adding H to each PGA, it creates molecules called G3P, also known as glyceraldehyde 3-phosphate. The next stage in our factory is called carbohydrate formation. This is where G3P is turned into carbohydrates, or sugars. Now, since glucose needs 6 carbons to form, because the chemical structure for glucose is C6H12O6, it steals 2 molecules of G3P. And finally, we move to the last stage of our factory cycle. It's called the regeneration phase. This is where G3P, with the help of ATP from our light energy reactions, is reused to make RUBP. Now, since glucose only needs 6 carbons, the byproducts of the reaction, or our factory waste, is recycled to make RUBPs. See, right here? And then it restarts the whole cycle. In summary, the Calvin cycle, or the Calvin factory, generates the exact number of elements to keep the process going. Now you might be asking, where was Ellie in this whole journey? Well, Ellie was being transferred between each molecule in the whole cycle. Now you might be asking yourself, what if the cycle was linear? Well, it would take forever to produce glucose. Having products reduced helps keep the flow stable. The factory reuses and rebuilds all their products, the same way we reduce, reuse, and recycle. The glucose that we produced from our Calvin cycle will now be used in the glycolysis process. Glycolysis literally means splitting sugars, and it takes place in the cytoplasm of a cell. There are 2 phases in this process, the investment phase and the energy harvesting phase. The process of glycolysis is a 10-step process which ends up with 2 ATP, 2 pyruvate, and 2 energy-carrying NADHs. Now we start with our glucose molecule, and with help from ATP and our enzymes, 2 AT molecules transfer energy to the glucose molecule to form fructose 1,6-biophosphate, right here. This is where the next stage begins, the harvesting energy phase. Now for this phase, fructose 6-biphosphate splits with our enzyme aldehase, and with more help of our ADP transferring into ATP and our 2NAD plus 2-pi transferring to 2NADH, we end up with pyruvate. In the next stage, pyruvate goes into the mitochondria by oxidation, and electrons are transferred from NAD to NADH. Ellie will have so much fun. And a carbon is lost in making carbon dioxide. It is then converted to a compound called acetyl-CoA. At the beginning of the Krebs cycle, you can see what I previously explained with the pyruvic acid turning into the acetyl-CoA. Acetyl-CoA bonds with 4-carbon molecule oxaloacetate within this cycle. Oxaloacetate is the beginning and the end of the cycle. The Krebs cycle is a series of redox reactions which are enzymatic. All carbons, hydrogens, and oxygens in pyruvate end up as carbon dioxide and water, as you can see by the blue circles in the cycle. Every glucose in the cycle has to go through the cycle twice in order to make 8 NADF, 2 FADH2, 2 ATP, and 6 CO2. And Ellie is found within those energy sources. On to the next stage of our journey, to the ETC, where we'll follow Ellie, who is an electron on the NADH. The electron transport chain takes place in the mitochondrial matrix and the intermembrane space. Within this process, it passes electrons through membrane proteins and takes energy to produce ATP molecules. Similar to the thylakoid membrane, the intermembrane space and mitochondrial matrix is separated by a phospholipid bilayer, which has multiple proteins layered within it. Proteins in the chain pump H+ ions across the membrane, while Ellie is being transferred between each protein. When the H ions flow back across the membrane through the ATP synthase compound, ATP is synthesized. O acts as a terminal electron acceptor by accepting electrons, shown right here. Oxygen is then reduced to form water, and Ellie is back with her hydrogen and oxygen friends. Thank you for following Ellie's journey. To recap, she started as a water molecule and was released during photolysis and entered into photosystem 1 and photosystem 2, where she created one cyclic phosphorylation before completing her journey as an NADPH. And we followed her through the Calvin cycle, where she became part of the G3P and then became a glucose. But we didn't stop there. Her journey wasn't done yet. She then became a part of glycolysis and then part of acetyl-CoA and entered into the Krebs cycle. And then from there, she turned into NADH and FADH2 and eventually went through the electron transport chain, ETC, and finally reunited with her hydrogen and oxygen friends.